The main objective of this research is to evaluate the existing strategies to provide precursors for the enhancement of secondary metabolite production in solid-state fermentation. The phenolic precursors can be synthesized using the shikimate pathway and the malonic acid pathway. Meanwhile, the terpenoid precursors can be synthesized using the mevalonic acid pathway and the methyl erythritol phosphate pathway. There are several problems commonly encountered in the production of secondary metabolites, in which one of them is the low yield of secondary metabolites obtained from plant extraction. This results into an overexploitation of plants, which poses a risk in harming plant biodiversity. Besides that, secondary metabolite production also faces a challenge of the influence of many culture conditions, such as the composition of culture media, the carbon-nitrogen ratio, salinity, the presence of metal ions, temperature, pH, and oxygen concentration. The existence of various factors complicates the improvement of secondary metabolite production. Aside from that, another problem commonly encountered is the specificity of the metabolite production. This is because some microorganisms only produce secondary metabolites in solid-state fermentation despite growing well in submerged fermentation.
Now we are heading to methodology. There are four main biosynthetic pathways of secondary metabolism, which are shikimic acid and malonic pathway, which will produce phenolic compound, while mevalonic and MEP pathway to produce terpenoid compound. For shikimic acid pathway, it have two ways. The first one starts with glucose going through the pentose phosphate pathway, producing the erythrophosphate phosphate, then going to shikimic pathway and producing phenolic compound. The second one is the same, but instead of going through pentose phosphate pathway, it undergo glycolysis, producing phosphoenol pyruvate, going into the shikimic pathway and producing phenolic compound. This pathway is essential for organisms that cannot obtain aromatic amino acids from their environment. Next is malonic acid pathway. From phosphoenol pyruvate, it produces pyruvic acid, which then later producing acetyl-CoA. This acetyl-CoA will go through malonic pathway and end up producing phenolic compound. This process catalyzed by acetyl-CoA carboxylase, and it is essential for cellular membranes and energy storage. The third one is mevalonic acid pathway. It's the same as malonic, but instead of going through malonic, it is going through mevalonic instead. The end product is terpenoids instead of phenolic. This process catalyzed by HMS-CoA reductase. It is provide building blocks which are important for plant defense and various biological activities in animals. Last but not least is methyl erythritol phosphate, in short MEP pathway. It derives from phosphoenol pyruvate to pyruvic acid, going through MEP pathway and producing terpenoids. This process catalyzed by 1,2-deoxydecellulose 5-phosphate synthase. It is essential for producing isoprenoid compounds in organisms that lack the mevalonic acid pathway. Last part of methodology is how SSF works. It works by growing microorganisms on solid substrate to produce the metabolites. It allows higher yield metabolites compared to the others. It is widely used and utilized selective precursor feeding together with metabolic engineering. When the rate of conversion in the pathway increases, the higher amount of the product will be produced.
The use of precursor to enhance secondary metabolite production has shown significant results in this article. There are 5 different types of precursor iodinase, such as tetracinomansin biosynthetic pathway, isoamyl alcohol, engineering, process supply chains and packaging, methionine, and the last one is isobentonylate phosphate and dimethynyl diphosphate. The first precursor is methionine. The addition of methionine to the culture media of Aspergillus strain favored locustatin biosynthesis during submerged fermentation, with similar effects observed in solid-state fermentation. The methionine added after 72 hours of incubation, which it will act as the locustatin precursor during the ideal test. Methionine supplementation in the solid-state culture of Aspergillus increases the locustatin production, which, emphasizing its role as a source of methyl groups in polypeptide chain biosynthesis. Second is isoamyl alcohol precursor. The addition of isoamyl alcohol as a biosynthetic precursor substantially increases isoamyl acetate, and a food additive with a banana-like odor 30 times higher, which these are observed in the submerged culture of Piscia fermentans. Thus, in submerged culture, production increased 30-fold, while in SSF, gradual isoamyl alcohol addition boosted production more than 12-fold compared to basal levels. This approach effectively minimized precursor toxicity while optimizing product yields. Third is tetrasinomycin biosynthetic pathway. Production of tetrasinomycin in Streptomyces cinnamonensis, an industrial monensin producer strain, was achieved by introducing the tetrasinomycin biosynthetic pathway. This strategy resulted in the production of 0.6 gram per liter of tetrasinomycin C and 4.35 gram per liter of tetrasinomycin A2 without affecting monensin production, demonstrating the strain's capacity to provide sufficient precursor for multiple biosynthetic pathway. Precursors are isopentenyl diphosphate and dimethylallyl diphosphate. The amplification of this precursor synthesis flux in the NEP pathway significantly enhanced lipopyrimidine production in E. coli. Incremental overexpression of bottleneck enzyme combined with reprogramming key metabolic nodes through protein engineering lead to a remarkable 2,600-fold increase in lipopyrimidine levels. Lastly, the last precursor are engineering precursor supply genes and AVEC gene. Engineering precursor supply genes and the AVEC gene in the industrial strain Streptomyces avermitilis greatly enhance avermectin B1A production. This integrated strategy achieves a maximum avermectin B1A production, which the percentage is higher compared to the unmodified strain. These studies highlight the effectiveness of precursor sublimation and metabolic engineering in optimizing secondary metabolite production.
Based on the article that we have chosen, there are 3 ways to enhance the secondary metabolite production in SSF, which is by stimulating the internal precursor generation, using precursors that are already contained in the solid material, and adding external precursor to SSF. The first method, which is stimulating the internal precursor generation, utilizes a 2-step supply energy to increase the production of tetramethylpyrazine, which is a flavoring additive with a nutty and roasted taste by Bacillus subtilis in SSF, and this resulted in the improved tetramethylpyrazine production when added with distiller's grain. In soybean seed capsules, Cosaconia cowani developed an exopolysaccharides that promotes plant growth. A combination of cane bagasse and broad bean seed capsules enhanced with sodium nitrate provided the highest yield. According to the scientists, sodium nitrate raised the expression level of proteins involved in exopolysaccharide production and exopolysaccharide synthesis. The second method is by using precursors that are already present in the SSF substrates to produce secondary metabolites. Substrates such as wheat straw and sugarcane bagasse may contain ferulic acid, which is a precursor to the formation of vanillin. Although there are no reports on its ability to make vanillin, Lindsay et al. Examined 9 agro-industrial byproducts in SSF and discovered that Penicillium camemberti produces it during the fermentation of olive cake. Other examples are such as GABA yield enhancement by Aspergillus oryzae and Monascus species through SSF using rice, and IV production enhancement in Astragalus membranaceus using Poria cocos SSF, which is mainly used for its wide health benefit. The third method is by adding external precursors to SSF to enhance the production of the desired compound. MSG, L-glutamate, L-phenylalanine, and isoamyl alcohol are examples of precursors that increase the formation of metabolites in SSF. To maximize yields, parameters such as pH, temperature, incubation duration, and substrate composition must be optimized. In this research, the scientists use volatile precursors such as isoamyl alcohol, as it can be gas phase fed to increase synthesis while reducing the toxicity.
Now let us discuss the three main ways to use precursor for secondary metabolite production by microorganism. Number 1 is modifying medium composition and fermentation condition. By adjusting its parameters can simulate the generation of metabolite. Changes in medium components can influence carbon flux distribution, leading to precursor accumulation. For example, adding glucose can enhance the synthesis of acetone. Studies show that altering the medium can induce transcriptomic changes, such as activating silent gene. Next, utilizing precursor already present in the solid substrate depend on the microorganism enzymatic capacity to convert the precursor, the concentration and bioavailability of the precursor in the substrate. For example, vanillin production that is from wheat straw, sugarcane bagasse, or olive card. The last one is applying external precursor can be added directly to the system with non-volatile precursor and volatile precursor. So how can we supply precursor to a solid-state bioreactor? Figure A shows that non-volatile precursor can be added directly to the culture medium by using a pump or sprayer on solid material, while Figure B shows that volatile precursor can supply through a bubbling column which humidify the extreme providing oxygen. Challenges in precursor utilization can happen when inefficiency arises in which precursors are redirected to an unintended pathway due to enzyme specificity and interaction with metabolite like acetyl-CoA. Bioreactor design depends on the precursor type and microorganism, like how stir drum bioreactor are ideal for yeast with beta heat and mass transfer. Scaling up challenges due to heat transfer is the main bottleneck, along with issues like mass transfer, moisture control, and aseptic operation.
In conclusion, optimizing the precursor pathways and the bioreactor designs is essential to increase the production of secondary metabolites and to reduce the use of energy and raw materials. The critical aspects which needs to be considered are the physical, chemical, and biological properties of the precursors used. Getting a thorough understanding of the precursor's method of interaction with the solid support and the culture medium is crucial. Another critical aspect which needs to be considered is the type of bioreactor used for the bioconversion of precursors. Choosing the right type of bioreactor can increase the bioconversion rate of precursors.